Hi, welcome back. Matrix, you need to pay very particular attention now because we're going to be focusing on meiosis. Now, before the break, we kept emphasizing that meiosis has two purposes. The one is to halve the chromosome number, because we can't take a cell with a full complement of chromosomes and another cell with a full complement of chromosomes and join them, because we'll have too many chromosomes. So we need to halve the number of chromosomes. Now, we can't just cut the chromosome in half or say, okay, that cell's going to have chromosome 1 and that cell's going to have chromosome 2 and divide them up like you deal out a pack of cards. There is a very simple way of doing this halving of the chromosome number. Remember, we said you've got homologous pairs. You've got a paternal homologue that you inherited from your father. You've got a maternal homologue that you inherited from your mother. And so a very simple way, because these two homologues are having the identical genetic information coded on them, a simple way is when you divide the cell and you're going to have half the number of chromosomes is to say, okay, this cell can have the paternal homologue and for chromosome 1, and for chromosome 2 it can have the maternal homologue, and for chromosome 3 let's give it the maternal homologue as well. Now, it's not a case of let's give, it's a completely random assortment of the chromosomes. And so you might have, on your paternal homologue, you might have had brown eyes, the gene for brown eyes, and on your maternal homologue you had also for eye color, same gene, but a different expression of it from your mother, blue eyes. And depending on what you pass on, which of the homologues you pass on to your offspring will help to determine their eye color. So we're going to start looking at this process of meiosis. We're going to look at a hypothetical cell that has 4 chromosomes, because if we started with 46 chromosomes, there wouldn't be space on this little picture for us to see everything. So we've got a hypothetical cell with 4 chromosomes, and we're going to say that there's 1 and 1, that's a homologous pair, and this big long chromosome, there's the other homologous pair. And just as with mitosis, just before meiosis takes place, DNA replication has to take place. And what we're therefore left with is this lovely long pink chromosome has replicated the DNA. It's held together with its little centromere. Same with the purple one and the little green one and the little blue one. They've now got carbon copies of each other. But what happens during prophase 1 of meiosis, you need to remember that with meiosis there are 2 nuclear divisions. In mitosis there was simply 1. In the first stage of prophase, everything happens identically to how it happened in mitosis, except we have homologous chromosomes coming and pairing with each other, lying next to each other. It's like they're sorting themselves out in neat little bunches together. And something hugely important happens, and that is the process called crossing over. Now, we said that one of the purposes of meiosis was halving the chromosome number. The other very important purpose of meiosis is to recombine the genetic information. You do not inherit all of your father's homologues, in other words, all the paternal homologues or all of your maternal homologues. It's a complete, what we call random assortment. In any gamete, in any given gamete, there's a complete reshuffling of the information on those homologues, and that happens in this process of crossing over. So here we have our 2 homologous chromosomes. Let's say this one comes from dad and this one comes from mom. A process where one of the chromatids leans over and we get a crossing over, so that when these two homologs move slightly apart from each other, we find that a little bit from this chromosome is now attached to that one, and a little bit from that chromosome is attached there. We have a new combination of genes. We've shown in this diagram that it happens once, but in actual fact it's happening multiple times along that homolog or along that chromatid. So if we have a look at our picture again, we can see that crossing over happens with those homologous chromosomes, and when we go into metaphase 1, we're going to see that these homologs are going to lie in a double row along the equator. In mitosis, it was just a single row. That's how you would tell the difference if you were given a test question and said, is this picture mitosis or meiosis? Well, you'd have to see if there was a double row, it's going to be metaphase 1 of meiosis. And in anaphase 1, the homologous chromosomes, the whole chromosome, is pulled up towards the pole. That chromosome does not split. And so what we have in our two daughter cells that are formed, we've still got 2 chromosomes, whereas in the original cell we had 4. After the break, we're going to look at meiosis division 2 and see where this takes us to the end of the meiotic division.